And we're going to be moving on to some translational activities. So the first speaker that we have is Dan Anderson. He's a colleague at the Koch Institute, also in the Institute for Medical Engineering and Science with me, and also in chemical engineering, many hats. Dan trained as a graduate student at UC Davis, where he studied DNA replication. He then came here to MIT, where he postdoced with Bob Langer. And he loved our environment so much that he's chosen to stay as a faculty member. And just this spring, he was promoted to tenure. So he's now a permanent and welcome colleague. So the title of his talk today is Intracellular RNA Delivery. Welcome, Dan.
Hello. Oh, great. Thanks so much for the invitation and chance to talk today. So I'd like to talk about a little bit of our work on intracellular delivery. And just as a preface, I think it's become increasingly clear that there are just an enormous number of biomolecules and drugs that could really change medicine, but they need to be delivered inside of cells. And so this is really a partial list. You heard from Phil today about the growing list of different RNAs, but there is also DNA and proteins. And so I think there's no question that these compounds have therapeutic potential. Really, the question is, how do we get them to where they need to be? And so I'd like to start off first by talking about siRNA, which is one of our favorites. So just to, again, kind of reiterate what Phil has described, the exciting part about siRNA is that it really does offer this opportunity to do what he describes as modular pharmacology. So really potentially having one drug that can eliminate a message to essentially any target and destroy a gene. And so clearly this has tremendous therapeutic potential, and the question is, how do we deliver it? So it turns out that RNA is really not the best drug by itself. So it's a relatively large molecule. It's highly charged. It doesn't naturally cross cellular membranes. It's sensitive to nucleases that are present in all of our cells. And it can even induce immune responses. And so that's really just one aspect of the challenges. Another is really the transport challenge. So if you're going to envision making a drug out of RNA, so first we need to basically have this RNA travel through the body, avoid non-target tissue. And once it reaches its target, it needs to extravasate and then actually enter cells. And that's typically done through endocytosis. But once inside the cell, it needs to escape that endosome. And even then, if it's an encapsulated form, it needs to decomplex to release the payload and perhaps even get to the nucleus, depending on what you're trying to do. And so it's really a multi-step process that's very challenging. And so these are really kind of the three key pieces in my mind of turning RNA into drugs. So the first is in some ways kind of obvious, but you need to be careful about the sequence of your RNA, because there are many potential sequences you could think about turning into a short RNA. There are really an amazing set of chemical tools now to facilitate delivery. And so there are tools to stabilize for nucleic acids. There are tools to reduce the immune responses that are natural to these kinds of molecules. There are even ways to attach ligands to these molecules to facilitate specific cellular uptake. And so I'm very fortunate to have John and Stan speak after me, and they're going to describe some of the really exciting advances in this area. And I'm going to focus mostly on encapsulation. So it's also possible to take an RNA and put it inside a nanoparticle. And using that nanoparticle, actually have the nanoparticle be the system that does the delivery. And this is really particularly important for larger RNAs that may not have as many tools available for them. So when I thought about doing RNA delivery, I was naturally inspired by all of the great work that's been done for several decades with cationic lipids, and this is really just a partial set of the molecules. And I don't want to get into too much chemistry here because this really is a biology symposia. Just to kind of give you a flavor of some of the challenges. DOTMA, this is the structure of a cationic lipid developed in the 1980s. And this is D-lin-DMA, which was actually published in Nature in 2006. And now they're really only different in a few places, and that's not to take away from the importance of D-lin-DMA and the advances that were made in this structure, but just to say that perhaps at least the materials that had been published when I started working in this field were not as diverse as perhaps they could be. And so one of the key questions was really first a chemistry question. How do we make large collections of relevant delivery molecules quickly? And so if you're a chemist, so Phil, you're actually a chemist, so back in the day, if you wanted to do this, this wouldn't actually be all that challenging. It might take you a couple weeks, but if you wanted to do this 1,000 times, and each time you did it, it was different and needed to be customized, it actually becomes a real challenge. And so really one of the first key strategies for our program has been to develop combinatorial methods. So coming up with the simplest possible strategy to make lipid-like materials quickly. And this is just one of the chemical methods we use. It's called a Michael type addition, and basically you can take an acrylate, as shown here, with an R group, and an amine, and a stir bar and a heater, and come up with a structure that in some ways is really lipid-like. So if you compare this structure to DOTAP, which is a conventional cationic lipid, you'll notice there are esters, there are tails, there are amines. And so we felt that this collection of molecules warranted further investigation. We've actually made thousands of these things now. And you can complex them with RNA. So it's actually not that difficult. You can take a cationic or an amine-containing lipid or polymer. You can combine that with colipids that may include polyethylene glycol, as you heard from Sangeeta, or other lipids like cholesterol. You combine it with RNA. And then you do a controlled mixing to put all of these things together and make nice nanoparticles of a range of different sizes. And 100 nanometers is just kind of a typical size that we get when we do these things. So if you have thousands of nanoparticles, really a key question is what are you going to do with them? And this was a real challenge early on when we could make more delivery systems than we could test. And so it was really Kinch at Alnylam, formerly from MIT, who developed this assay. And basically what he figured out was that you could take a factor 7 siRNA, so this is an siRNA targeting a blood clotting factor, make a nanoparticle, and then do a simple injection in mice and just measure in the blood whether or not that blood clotting factor was reduced. And so now instead of really having a lot of trouble testing our particles, we could go about testing literally hundreds of different formulations quickly with the system he developed. And just to put all of this together, the basic idea is you try to come up with novel chemical strategies to make new materials to make your nanoparticles out of. You try to automate these systems when you can to try to facilitate doing the chemistry. We do controlled mixing. We often use a microfluidic system to really be as precise and also to miniaturize the systems. And then we test these in animals to see if they work. And the bottom line is this process really does work. So the first data I'm going to show is actually describing the target TTR, which John is certainly going to tell you a lot of exciting developments. But just for the purposes of our study, it's an example target in the liver in hepatocytes. And so we made a nanoparticle with our new material. We inject this into primates, and then we measure whether or not we can get knockdown in hepatocytes. And the bottom line is that these things work. So even at low doses, relatively low doses of RNA, once you formulate them, you can see very significant knockdown in primates. And this is just one example of this dose of RNA, that you're getting very good knockdown in primates. This approach allows us to develop a lot of structures that allow us to do sort of next generation structure function studies so we can get smarter at making nanoparticles. But overall, this process has really been amazing over the last several years. This is data from Alnylam and also from Peter Cullis and Alcana. This is really kind of a composite of work from many people showing the progression of the potency of RNA delivery systems, siRNA delivery systems over the years. So back when everybody first started, this was really the most potent delivery system. So what you're looking at is knockdown of the factor 7 target and the amount of RNA it takes. And so here you can see these first systems take 10 milligrams per kilogram of animal, and over the course of the years, they become more and more and more potent, really multiple logs more potent. And we've participated along the way, and in fact, recently this year, Yizhao and our group published what we understand to be, at least publicly, the most potent system, which is giving us knockdown at 0.002 mg per kg. So just an incredible increase in the potency of these delivery systems, and sort of showing that if you work at it, it really is possible to make better nanoparticles. And so because these are very potent, you can actually multiplex them. So you can take 10 different RNAs to 10 different targets, make a nanoparticle that encapsulates all 10, and then when you inject these into animals, you can see knockdown of all 10. And so this is an example of that, where if you take the nanoformulation and you look at knockdown, you can see all 10 of these targets, not at an equivalent level, but you do see significant knockdown in all of them. And so now we can think about making drugs that really don't just hit one target, but may hit more complex targets, as is certainly common in cancer. So I thought I'd spend a little time talking about the fact that this type of delivery system isn't limited to hepatocytes. I think there's a perception in the field, or at least in the business community, that this is, the progress has been phenomenal and great, but this is only going to work in the liver, and I think that's simply not true, and you heard some of that from Sangeeta. I want to give one example of our work where we've been developing systems to deliver siRNA to the endothelium, which is an important target. And so, again, at a high level, this approach is really still the same. So we develop methods for making large libraries of delivery materials. In this case, they're short polymers. We do controlled mixing to make nanoparticles containing RNA. And then now, instead of testing whether or not they deliver to the liver, we're testing whether or not they can deliver to blood vessels, the endothelium. And this is work recently published. James Dahlman was the lead inventor on this. So you can take these materials and combine them with some colipids and siRNA and make nanoparticles. In fact, this is a nice EM picture showing how, if you look closely, they almost look like jawbreakers with multiple layers of lipid and RNA. This is a very reproducible system where you can make the same nanoparticle over and over again, and it can be stable for many, many days for storage. So if you take a nanoparticle made out of this material with an RNA targeting ICAM2, which is common in a lot of endothelial cells, inject it into a mouse and then look at knockdown, you can see knockdown in a range of endothelial cells. And so in this experiment, what we're looking at is knockdown of ICAM2, as shown here, and a decreasing dose of RNA. So at the highest dose, you're seeing knockdown in the lung endothelium, the heart, the kidney, the liver. And as you decrease the dose, you notice you're still getting silencing, but in particular, we see a lot of silencing in the lung. And so this system in particular seems to be very potent at delivering siRNA to the endothelium of the lung. And again, because it's so potent, we don't just have to think about delivering one siRNA, we can think about delivering many. And so this is an experiment where we did 5 different siRNAs to 5 different targets, make our nanoparticle, inject the animal, and then look at knockdown in the blood vessels of the lung. And what you can see is at all of the doses that we tested, we see knockdown in all 5. And so this is looking at knockdown of 5 different targets, Tie1, Tie2, VEGFR2, VE, cadherin, and ICAM, and a decreasing amount of RNA. So again, at all of the doses tested, we're seeing significant specific knockdown in our target genes. And so one of the sort of interesting features of this delivery system is that changing the chemistry to develop a endothelial delivery system also changed the tropism. So if we compare, just do a simple comparison looking at knockdown of our previous liver delivery system, if we look in the lung with our last delivery system, we see no knockdown at any dose. We just can't get it to work in the lung endothelium. This new system, even at very low doses of RNA, we're seeing knockdown in the lung. In contrast, if we look in the liver, our previous system is very good and potent at delivering in the liver of hepatocytes, and the new system is very poor at delivering to hepatocytes. So there's something that we've done by changing the chemistry of this nanoparticle that really changes where it goes and where you get knockdown. So naturally, being in the cancer center, we're interested in whether or not these systems can be used with cancer. And of course, one of the most famous mouse models in our building is certainly Tyler Jack's lung cancer model. And so in collaboration with Tyler and his graduate student, Wen Zhu, we were curious to know whether or not it was possible to use our lung-targeting RNA delivery system to treat lung cancer. And so this is just an example of the system. So this is a Cre-mediated KRAS P53 model system of non-small cell carcinoma. So the idea here is to basically affect the biology specifically with these nanoparticles. So can we specifically knock down KRAS with a KRAS siRNA? And can we affect p53 indirectly by delivering a microRNA? So we're not actually delivering p53, but we're delivering a microRNA that is involved in the p53 pathway. And so we make our formulation with these two different siRNAs, 1 or 2, and our new material and our polyethylene glycol lipid, and we make our nanoparticle. And then we ask the question, can this nanoparticle deliver and silence in tumors? Can it affect tumor growth? And can it ultimately be used with other drugs to treat the cancer? So first of all, for delivery, this is an example of I don't know how clear it is, but these tumors express luciferase in tomato red. And when we use the luciferase targeting siRNA, we see silencing in the tumor cells themselves after administration in vivo. And so now if we look at tumor growth, if you use a microRNA delivery nanoparticle, you can see a very significant reduction in the rate at which these tumors grow. So this is basically making the tumor-containing mice, injecting the nanoparticles, and then measuring tumor growth over time. So miR34A will dramatically reduce the growth rate of the tumors. KRAS will similarly cause a reduction. And actually, if you combine the two, you see a combinatorial effect where you actually start to see regression of these tumors in vivo in this very tough model of lung cancer. And finally, you don't only have to use siRNA. One of the mainstream therapies in this model system is cisplatin. And so you can combine cisplatin and the nanoparticles with the different siRNAs. And in this experiment, you're basically looking at survival of the animals following a series of injections of different combinations of drugs. And so with just the control formulations, you see this type of survival curve. With either cisplatin or the combination RNA therapy, you see an increase past the last injection. And with both, you see an even larger increase past the last injection. So we're excited by the potential of these types of nanoparticles to be used to treat this tough disease of cancer. There are other cells that we think are accessible with siRNAs, so immune cells. Now, there are many different immune cells, and not all of them are amenable to siRNAs. But in particular, it turns out that some of these leukocytes, dendritic cells and macrophages, can be hit with existing nanoformulations. And so here's an experiment we did in collaboration with Alnylam, where we took a CD45 RNA or a control RNA, injected it into non-human primates, and then measured peripheral blood leukocytes following the therapy. And what you can see is you see a very significant reduction in the treated group of CD45. So it's specific to the RNA, and you really are hitting the majority of these cells in the peripheral blood. And this system has been used with different RNAs and different targets and even some therapeutic models, but I think it's clear that at least in model systems, we can certainly hit certain cells in the immune system with RNA. And so right now, just to give some perspective on where we are with delivery, we can deliver to all the cells in the liver, and in fact, you're going to hear some exciting human hepatocyte data from the following speakers. But we can also hit Kupffer cells and stellate cells in the endothelium. We can hit the endothelium of many organs, including kidney and liver, spleen, heart, and lung. We can hit certain leukocyte populations, and that's been shown in primates. And we can also deliver to a growing collection of tumors, including some of those that you heard from Sangeeta earlier. So I wanted to speak very briefly about just some of the transport aspects of nanoparticles. So what actually happens inside of a cell when you deliver a nanoparticle? And this is actually kind of a complicated question that involves a lot of pathways and discussion, but I only really wanted to focus on one key piece, which I think is interesting, which is that it turns out that if you look, at least in vitro, I always used to think that once the RNA gets in, it stays in. But in fact, in our systems, when you add nanoparticles with siRNA, actually about 70% of it is recycled out again. So there's a process of endocytosis, but there's also a process of exocytosis. And in fact, in cell culture, what we see is the majority of RNA is taken in and then kicked back out again within a day. And so, we were thinking, well, perhaps one strategy to improving potency is actually to manipulate these cellular trafficking pathways. And there's one pathway in particular that's known to be important in cholesterol recycling and metabolism, Niemann-Pick type 1. It's actually associated with a very nasty genetic disease as well. But it turns out that in NPC1- cells and even animals, you see a dramatic difference in how these types of nanoparticles accumulate and function. So this is a picture showing in the wild-type cells the amount of labeled nanoparticles that you see in the cellular area. And this is an example in the mutant cells, how you get this dramatic accumulation of lipid nanoparticles and labeled RNA in cells. And this actually, recently we've shown this holds true also in vivo. And so, number one, it's interesting to start to understand the key features of endosomal and excitosis of nanoparticles, but also to think that perhaps there are ways we could interfere with these processes to improve delivery across the board. This was recently published by Gaurav Sahay and ourselves last year. So what about other types of RNA? So really, in my mind, the other half of modular pharmacology is turning genes on. So I think it's very exciting to think about turning genes off with siRNA, but certainly it's clear that you're not going to be able to treat every disease just by turning something off. And so there's been this very rapid expansion and interest in thinking about delivering messenger RNA, where now you deliver the message to a new protein inside of cells to turn genes on. And so when we think from a delivery perspective what the challenges are, so siRNA is largely double-stranded, which provides another layer of protection from nucleases, while mRNA is largely single-stranded, making it more sensitive. There are an amazing collection of chemical tools available to manipulate RNA and RNA analogs doing conjugation. Again, you're going to hear some of this next. But mRNA really does have a much more limited set of tools. So it's not so easy to just make large pharmaceutical quantities of exactly which type of mRNA you want with all these stabilizations and modifications. And while siRNA is big, mRNA can be really, really big. So this is an enormous molecule. And at least today, it looks like the approaches with the most promise involve encapsulation until we can perhaps have some of these chemical tools catch up. So it is possible to take a messenger RNA or a modified messenger RNA and a lipid nanoparticle and combine that with various colipids and do controlled mixing in a very similar way to how we do our siRNA nanoparticles and make some encapsulated forms. And we first started actually looking at cellular transfection. So we were interested in developing delivery systems to deliver mRNA inside of cells in cell culture for reprogramming purposes. It turns out actually now there's a kit sold based on some of our materials. But in this process, it became clear that the amount of expression we could see in cells was really dramatically higher than what we were getting with our DNA delivery compounds. So in fact, really in many different cell systems, we can see essentially nearly 100% transfection with mRNA. So this is an experiment where we're delivering a green fluorescent protein in a nanoparticle. So the first in vivo data that I want to describe is actually delivering a protein called erythropoietin. You're probably familiar with EPO. You may be familiar with Lance Armstrong and EPO. But the bottom line is it's a very important therapeutic protein involved in treating anemia. So it induces red blood cell production. And so if we take an mRNA encoding EPO and we make our nanoparticle, inject a mouse, and now we're delivering to the liver, but we're measuring soluble human protein. So EPO is delivered to the liver, you have new protein produced, and you can measure the human protein in the blood. And so this is done in collaboration with Shire Pharmaceutics and Brad Guild, who's actually in the audience, and Mike Hartline and others. So what you see is after you give your injection, you see this very rapid increase in the physiological levels. In fact, the amount of human EPO that we see in the animals is about 60,000 times the physiological level. So it's just an enormous amount of therapeutic protein delivered. You see a corresponding increase in hematocrit. The amount of red blood cells becomes so large that the blood becomes kind of viscous and dangerous. But this is really just a demonstration of the fact that you really can get enormous levels of protein circulating following delivery of a nano-encapsulated system. In fact, this has now been also demonstrated in primates. These same formulations are working in primates and are actually also in clinical translation today. Here's one more example of an mRNA payload. This is factor IX. So factor IX, I'm not going to walk through the pathway, but it's involved in blood clotting. So just really demonstrating the modularity of these systems, we take a different mRNA to a different protein, but everything else is the same. And so we take our mRNA encoding factor 9, inject it in mice, look at soluble protein. Sorry, that's not EPO, that's factor 9. And again, we can see very large levels of factor 9, showing potential to use these types of formulations to treat hemophilia. In this case, we're getting about almost 100 times as much as above the hemophiliac level, so very large amounts. So there are other aspects of modular pharmacology that are exciting. In fact, you're going to hear from one of the leaders in the field of the CRISPR technology, and I think CRISPR is a very exciting tool today to actually think about doing genetic repair inside of cells. So taking the CRISPR system, delivering it, and actually changing the nucleic acid so that you could perhaps fix a genetic disease. And there are other tools to do this too, talons and zinc fingers. There's a collection. But so the idea now is perhaps you could give someone one treatment and then they're fixed for the rest of their life. So I don't want to go into the biology because we're going to hear from, again, one of the leaders in the field. But from a delivery perspective, there are really kind of three things that we need to deliver. So when we compare siRNA, which is our short double-stranded RNA, this is actually much more complicated. We need to get a protein inside of cells. We need to get an RNA inside of cells, which is called the guide RNA, that targets it to the specific spot in the nucleus, in the DNA. And if we want to do repair, we also need to deliver a repair template. So we basically need to figure out how to get everything inside of the nucleus of cells. So we recently demonstrated and published this year, this was Hao Yin's work in our lab, showing that this type of system can be used in adult animals to treat a model of tyrosinemia. So this is a genetic disorder that leads to accumulation of FAA, it's an analog, and leads to liver and kidney damage. And so what Hao Yin and Wen and actually Phil and others, we all showed was that you could give a high-pressure injection, which is essentially, the reason this needle is so big is to really demonstrate that this is a lot of volume. So you're actually injecting 10% of the body weight of the animal very quickly over 5 or 6 seconds. So it's not a preferred method for treating humans, obviously, but it really is a very powerful tool for looking at model systems. And actually, I think there have been clinical trials. It's interesting to think about whether or not you might think about using this if you had to do it once and it cured a person. I think that's an interesting question. But anyway, what we showed was that you could take a plasmid encoding Cas9 and a guide RNA, and then a double-stranded repair template, and you could actually see correction of this disorder. So I won't go into the biology so much, but just to say you actually can see repair of the liver. You can see gain of function. You can see a loss of the damage associated with this disease. And so it really is therapeutic. Again, the challenge is, number one, the repair rates were kind of low. So we see only about 0.4% repair in this model system, which isn't going to be used to treat everything. But it's proof of principle that this really can work. And again, when we think about developing a clinically suitable system, the challenge is we need to deliver all of these systems. And so, unfortunately, the Cas9 protein is large and really doesn't fit into the current clinically suitable viral systems. It has been shown to be used in adenovirus, but there are some concerns about that. But it turns out that if you really think about it, you can combine an mRNA nanoparticle to deliver the protein. You can use a guide RNA in the form of a particle or a virus, and you can potentially deliver the DNA in a virus. And actually, we've just done this, and the correction rates are over 1%. This is really just data that's very new, and we think we can do much better. But we're excited because we think this is the first demonstration of a clinically suitable formulation to deliver CRISPR in adult animals. I think in the interest of time, I just want to end early. And thank a lot of really smart people. So I've been very fortunate to work with a lot of really smart people at Alnylam, in particular Kin and Carmen and Victor. Very large list of smart people at MIT. I've mentioned James and Hao and Roman and Yizhao, Tyler. We could go through the list, but there are a lot of really great people, in particular Bob Langer too. And then finally, Shire, including Brad, on some of our mRNA work. So thank you very much.